Good morning, everyone. In this talk, I will try to highlight some of our recent work on developing a new technique for identification of quantitative resistance to Blackleg. For many years, the field surveys on the prairies have generally shown that Blackleg is quite common, although the disease levels are mostly low, except in certain isolated cases. This is generally attributed to broad use of resistant cultivars, including both major gene resistance and possibly quantitative resistance. Interestingly, for many years, only limited number of R genes have been used in Canadian canola cultivars. They're mostly RLM1/LEPR3 and RLM3, although additional R genes are being introduced in the newer varieties in the last couple of years. Looking from the pathogen side, the avian genes that help trigger the resistance of those common R genes in our variety have been mostly at low levels in the pathogen population. Since 2010, when our monitoring started, so that means the resistance we observed with most of our varieties may not have much to do with the R genes in them. Instead, there could be some other factors in the play. We also notice most of our varieties seem to carry a level of a QR. In addition to the common benefit, QR seems to be particularly useful in Western Canada, and because we often would have a lower level of disease pressure than in many other regions. However, identification of a QR is not an easy thing because of the complex genetics of it. And industry mostly relying on extensive field trials to select a QR, which is time-consuming and sometimes with variable results due to the impact of the environmental conditions to the QR traits. If you have a lot of candidates to evaluate, and that can be a very inefficient process. So we're looking at a potential new method in this study to be able to quantify QR more efficiently for selecting breeding lines with the best QR traits or label QR in canola varieties. First, we look at the optimal inoculation method that would lead to the high incidence of stem infection. It seems that at the seedling stage, inoculation of a cotyledon will most likely lead to the stem infection as opposed to the inoculation of other lower leaves. Even between cotyledon and the first leaf inoculation, there is quite a bit of a difference in terms of disease incidence and the disease severity it can cause. At the same time, even with the cotyledon inoculation on a susceptible cultivar West Star, there was never 100% of a disease incidence it can cause. So there seems to be a barrier that reduced the infection um, development from infected cotyledon into the stem. So that's something we wanted to keep in mind when we develop the protocol. We also compared the cotyledon inoculation with the pedio inoculation, and it was apparent that the pedio inoculation would cause a higher or more severe disease than cotyledon inoculation did, measured by the plant mortality over a period of time. It was likely that the pedio inoculation would be able to bypass whatever the barrier is between the cotyledon infection leading to the stem infection. And also on the varieties with the different level of the resistance here, we refer to as a quantitative resistance. There is a clear difference among these varieties, especially with the CCC1 that seem to have a strongest resistance with both the cotyledon and the pedio inoculation. So it seems that using cotyledon or pedio inoculation, we would be able to differentiate different levels of quantitative resistance among canola varieties. So can we quantify QR with cotyledon or pedio inoculation? Often the modes of action for QR are different from those of major gene resistance. For example, with the CCC1 program, the cell death seems to be fairly important, which is not a strong one in terms of defense and can't stop the infection completely. But it does help to reduce the spread of fungal hyphae from the infection site and also reduce the expansion of the lesion on cotyledon. So it seems that the growth of the pathogen was quite restricted with this quantitative resistance. And we maybe termed it as the reduction of fungal growth kinetics, which helped reduce the infection progress from infected cotyledon into the stem. And before that happens, and because of the slowdown of the infection, the cotyledon might have dropped. So the question is, can we quantify the different levels of fungal growth kinetics in canola tissue, especially in relation to the different levels of quantitative resistance? We explored extensively our use of droplet digital PCR to quantify the pathogen growth kinetics in canola tissue. ddPCR is a powerful molecular quantification technology, and it seems to be fairly effective in this case. 2 weeks after cotyledon inoculation, the amount of pathogen DNA in pedio and stem tissue seemed to be substantially reduced or less in these resistant varieties as opposed to Westar. And this amount of a reduction in the pathogen DNA seems to be related to the disease resistance level with these varieties. And this is quite exciting. So we wanted to expand this study to include more the varieties for this testing. In further experiments, canola varieties or breeding lines with a range of resistance based on multi-year field data were provided by breeding companies. Mind you, the results from these field trials may involve both major gene resistance and QR because the disease was mostly caused by natural inoculum. And also these varieties carry only the common R genes as described before. Both the PDO and cotyledon inoculations were used, and with the pathogen inoculum carrying no avirulence gene 1 or 3 to avoid the major gene interaction. To quantify the fungal DNA in the stem tissue, samples were collected just below the scar of inoculated cotyledon or PDO. ddPCR quantification of a pathogen DNA was relative to the amount of DNA of a canola. Okay, after initial ddPCR assay, 14 of the varieties were selected for field inoculation trials based on both cotyledon and the PDO inoculation. And for a specific validation of a QR, because we can use inoculum to avoid the major gene interaction in these trials. The first part of the results shows the comparison of ddPCR data from PDO inoculation versus multi-year field results over 48 varieties. Both sets of data were expressed in percentage of a wet star. That means the lower the bars, the stronger the resistance. When paired together, the two sets of data seem to match quite well for most of the varieties, although there are some exceptions. This regression graph shows the good correlation between the two sets of data for PDO inoculation, with the exceptions where ddPCR seem to overestimate or underestimate the resistance levels as opposed to the field trial data. Similar comparison was also carried out with the ddPCR data from cotyledon inoculation with the field results over reduced the number of varieties. Again, the correlation was still reasonable, also slightly lower than the PDO inoculation. Additionally, the results of PDO and cotyledon inoculations seem to correlate each other, which is a good news because we would prefer to use PDO inoculation if it's possible, because it can produce almost 100% of a stem infection, which is useful because the ddPCR quantification of the pathogen DNA would use the stem samples. In inoculated field trials for validation of QR with the selected varieties, PDO inoculation apparently caused a higher level of disease than cotyledon inoculation, which is not surprising due to the position of the inoculation. When we looked at the disease incidence, there seems to be very little difference among these varieties, which is also understandable. And because of the inoculation position so close to the stem, which will cause a higher success of the infection of the stem. There's one variety seems to reduce the disease incidence quite significantly. That's a bit interesting. For disease severity index, there was a clear pattern of a different resistance measured by PDO inoculation and the cotyledon inoculation. And the pattern seemed to be similar between the two inoculation methods. This regression graph clearly shows the correlation between cotyledon and the PDO inoculation in these field trials based on the disease severity index. This table provides a summary of resistance performance of 14 selected varieties in different trials. It's a bit busy, but let's look at the resistance in the multi-year field trials. The susceptible, resistant, and highly resistant reactions were well reflected by the cotyledon inoculation in both ddPCR quantification and also inoculated field trials based on the disease severity index. However, for the PDO inoculation, this results of multi-year field trials can be well reflected by the ddPCR testing results only. But in the field trials, it seems that the PDO inoculation will induce a higher level of stem infection or disease severity than the other the assays can indicate it. So this is something to keep in mind with the inoculated field trials. To look at it a little bit more critically based on the regression or correlation, the ddPCR data with these selected varieties correlated well with the inoculated field trials based on the disease severity for only cotyledon inoculation, but not so much with the PDO inoculation due to the reason we discussed earlier. To sum up, quantitative resistance seems to be able to affect the kinetics of fungal growth in canola tissue, which can be measured with the ddPCR based on the amount of pathogen DNA. These ddPCR data are well correlated with the multi-year field results for blackleg resistance, as well as to the inoculated field trial results, specifically for quantitative resistance assessment. For application, I think this ddPCR assay may be used to screen canola germplasm or breeding lines for strongest or weakest quantitative resistance, especially when you have a large number of candidates to be assessed. This is a good approach um, to select the targeted candidates for field trials if that is the last step of the assessment. For QR labeling of canola varieties, it may need a little bit of more work, and we are expanding the assessment with the more commercial varieties. Just to acknowledge people who participated in these studies, uh, seed companies that provided the varieties and the field data, and also funding agencies that supported this research. Thank you all for your attention.